Hi everyone, so today we're going to cover microbial genetics and metabolic regulation as well as some eukaryotic facts to kind of help you distinguish the difference between genetic events in prokaryotes such as bacteria versus eukaryotes such as us. So the first thing we have here is just a reminder of the nucleic acids. And you'll notice that there is this little area here with the pyrimidine and the purine bases that are part of nucleic acids. And the trick to remembering which is cut pie. So like you would cut a pie, think C, U, and T are all pyrimidines, whereas A and G are your purine bases. And the difference between them you can see is pyrimidines are 1 ring, whereas purines are 2 ring structures. Okay? And it's kind of funny because the bigger word is the smaller structure. Okay? And then we also have here for you to see the phosphate sugar alternating backbone of DNA and RNA. And then here the full structure where you can see the phosphate, sugar, and nitrogen bases. Now on this slide what we're going to focus on is the gyrase or topoisomerase, which is an enzyme that you find in prokaryotes but not eukaryotes. And that's very important because since gyrase is involved in coiling and uncoiling DNA in bacteria, which is very important for their DNA replication, but it's not found in our human cells, in microbiology, that makes gyrase a very good target. You're able to kill bacteria if you inhibit gyrase. And you don't hurt the human because, well, we don't have gyrase. Okay, so make sure you know what gyrase does, which is coiling and uncoiling DNA. But also make sure you know that it is a good target to kill bacteria. Now, when we talk about genetics, one of the important things to always remember is what is the central dogma of biology. So anytime you see the term central dogma, I want you to write down DNA to RNA to protein, meaning that the way that genetics flows in biology is DNA is transcribed to RNA, which is then translated to protein. Now what I have here is just a little figure for you to see the processes that we're going to review today. The first one over here shows you DNA replication, and then you have that DNA being transcribed into RNA over here, and then you have that RNA getting translated into protein here. So this figure here nicely demonstrates the central dogma of biology, going from DNA to RNA to protein. Now, before we get into the details of each of the processes, I just want to show you a couple of videos to help you visualize and remind you what's going on in each of these processes. Because at this point, you should have encountered them in other classes as well, so we won't spend too much time going into the details of each one. So first up, we have a general overview of DNA replication.
In order to pass genetic information on to its offspring, an organism must make a copy of its DNA. The process of copying DNA is called DNA replication. During replication, each strand of the parental DNA serves as a template in the creation of new DNA. Because each newly synthesized DNA molecule is made up of one parental strand and one new strand, DNA replication is described as semi-conservative. One strand in each molecule is conserved, while the other strand is newly replicated. In many organisms, the two DNA polymerases responsible for replicating the leading and lagging strands are linked together by connector proteins. To make this possible, cells take advantage of the flexibility of DNA. The leading strand is synthesized continuously toward the replication fork in the 5' to 3' direction. Since the leading strand is synthesized continuously, it only needs one RNA primer to start the process. Since DNA polymerase can only add nucleotides to the 3' end of the growing DNA molecule, the lagging strand is synthesized discontinuously away from the replication fork in the 5' to 3' direction. The DNA of the lagging strand is synthesized in pieces called Okazaki fragments, which consist of about 1,000 nucleotides. Each Okazaki fragment requires its own RNA primer.
So as that video reminded you, when we talk about DNA replication, you're producing two DNA strands that are identical copies from an original double-stranded DNA. Now the term you have to remember for this process is semi-conservative replication. So ask yourself, what does it mean if it's semi-conservative replication? Well, it means that the original DNA is partially conserved, because in DNA replication, both strands will now serve as templates for the new DNA strand. So each of the daughter DNA molecules will have one strand that's the old original DNA, and a second strand that is then the newly built molecule. So that's why it's called semi-conservative. The other thing that you always want to ask yourself is, when does a cell need to replicate DNA? And we touched upon this in the previous lecture as well. A cell needs to replicate its DNA before mitosis. Okay, so always write that down, put stars, circle that. A cell needs to replicate DNA before mitosis, because if you think about it, if a cell is going to split, if it doesn't double its DNA first, then each of those new cells is only going to have half of the genes that they're supposed to. Okay, so it's very important that cells double their DNA first, before mitosis. So now to recap the specifics of what actually occurs in DNA replication, and for any of the genetic processes, if you understand what each of the enzymes does, then you would be able to explain the process to someone. So with bacterial DNA replication, the first two things you need are gyrase and helicase. We just mentioned a few minutes ago that gyrase is responsible for uncoiling the DNA that's currently all coiled up tightly in order to fit inside that little bacterial cell. Once gyrase uncoils the DNA, then helicase unzips it, meaning it denatures those double strands into their single strands, so that each strand will now be able to serve as a template. Now, when you have those double strands separated into single strands, any single nucleotide or nucleic acid basically is very single. and clingy and very much wants to find its partner. So what happens is, if you don't do something to prevent it, those single strands would stick right back together. So that's why the cell needs single strand binding protein, or SSB. Single strand binding protein you see in this diagram here. Once the two single strands are opened up, SSB coats each one to prevent the single strands from sticking right back together. Okay? Now we're going to move to the next slides to then explain the final enzymes that you see listed here. Once the strands are separated, now you can have polymerases come along. And anytime you hear the term polymerase, I want you to think an enzyme that builds the nucleic acids. So a DNA polymerase will build DNA strands, whereas an RNA polymerase will build RNA strands. Now, DNA polymerases have their limitations. So they're not that great at doing their job of building DNA. Their limitations are that they are one-directional and they can't start new strands. And when we say one-directional, that means that a DNA polymerase can only build a DNA strand from 5' to 3'. Okay, and remember, that's simply how we label DNA orientation. 5' to 3' is the only direction it could go. So that makes us wonder what's necessary to compensate for these limitations. If DNA polymerase can't work in any other direction and can't start new strands from scratch, how does new DNA get made? Well, that's where primase comes into play. Primase is a special RNA polymerase that will make little RNA primers, which you see in the figures here. Little RNA primers that will serve as a starting place for the DNA polymerase to then build off of. Okay, because RNA polymerases do not have limitations. They can start a new strand completely from scratch. They don't need any other base to build off of. So when you think of the order of enzymes in the process of DNA replication, we had gyrase and helicase, then we had SSB. Now we have primase comes along and puts the RNA primers, and then DNA polymerase builds the new DNA strand off of that little starting primer. Now that then leaves you with a problem. At the end of DNA replication, well, now you still have these little pieces of RNA in the double-stranded DNA. That's a problem because, as you already know, RNA has some different base composition than DNA. So RNA will have uracils in it, and it will lack any thymines, which would be a problem. So at the end of DNA replication, you need pol 1, RNase H, and ligase. What happens is pol 1 and RNase H, they degrade the RNA primers, meaning they get rid of all of the RNA bases. Pol 1, which is a polymerase, then replaces those bases with DNA bases. The last thing to happen is ligase, which serves as a little glue, will fill in any gaps in the backbone. So the ligase will basically glue in the new DNA bases. Okay, so again, what happened was the RNA gets removed, it then gets replaced with DNA bases, and ligase then does the final gluing of the backbone to fill in the gaps. Okay, so let's recap that one more time. The way DNA replication works is, first you have the gyrase, and the gyrase uncoils that big ball of DNA. Then the helicase comes along, and that unzips the double strands so that there are now two exposed single strands. Then SSB comes along and coats both of those single strands so that they don't instantly stick back together. Now that those strands are nice and separated, primase can come along, and that's the one that we said is a rock star. It's an RNA polymerase that will start off our new strands, but it's starting off as RNA bases, just a few, usually about 6 to 9 bases. And now that those bases are there, DNA polymerase, which has its limitations, now has something to build off of as a starting point. So now DNA polymerase can take over and build the rest of the DNA strands, putting together those A's, T's, G's, and C's. Once that's complete and we have our 2 new DNA strands, there's currently a problem, right? After DNA synthesis, there's that problem that there are still some RNA bases in that DNA strand because the primers are there. So now PO1 and RNase H will degrade or remove any of those RNA bases, and PO1, which is a polymerase, will now build or replace those RNA bases with DNA bases. The last thing to happen is ligase basically cleans up the last minute messes and will reseal any little gaps that are currently in the backbone of the DNA. And boom, there you have the bacteria has 2 new DNA strands, which are semi-conservative. They are made up of 1 original DNA strand and 1 newly built DNA strand. And all of this occurred before mitosis, okay? Before the cell needs to divide. So that after mitosis, each cell will have the proper amount of DNA, okay? If you have any trouble with any of the enzymes or any of the DNA replication process, just send me a remind message and I'll give you a bit more information. Again, this is meant as a chapter in microbiology that is supposed to be review, but I understand that some students may not have had this before. So feel free to ask as many questions as you like. You are never bothering me. So now we've reviewed DNA replication, and now the cell has divided. It's nice and happy with the right amount of DNA. Now it's time to actually use that DNA. And whenever you see the word express in genetics, that simply means to transcribe. So we're going to go through transcription now so that the DNA that's been replicated can now serve as a template for RNA.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins. Cells use 3 types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes. There are three stages in the process of transcription: initiation, elongation, and termination. Let's see how each stage works. During initiation, RNA polymerase binds to the DNA and recognizes a site called a promoter at the 3' end of the template strand of the target gene. When RNA polymerase finds a promoter, it breaks the hydrogen bonds holding the DNA strands together at the site of the promoter, and transcription begins. RNA polymerase does not bind to all promoters with equal affinity. This difference in promoter strength is one way that cells can control gene expression. The more strongly RNA polymerase binds to a particular promoter, the more likely that gene is to be transcribed. In general, higher levels of transcription lead to higher levels of translation, which leads to higher concentrations of that polypeptide. One strand of the DNA serves as a template strand. The RNA transcript is copied from this strand of the gene and therefore has an RNA sequence complementary to this strand. During elongation, RNA polymerase moves along the DNA, opening up a bubble in the DNA. As it moves, RNA polymerase adds bases to the 3' end of the growing RNA transcript. Elongation continues until RNA polymerase reaches a site on the DNA called a terminator. At this site, RNA polymerase and the newly synthesized RNA transcript are released from the DNA. Termination can occur through two mechanisms: self-termination and enzyme-dependent termination. In self-termination, the RNA sequence transcribed at the terminator causes the RNA to hydrogen bond with itself, forming a stem loop structure, which essentially pulls the RNA polymerase off the DNA. In enzyme-dependent termination, a termination protein binds to the terminator and pushes RNA polymerase off the DNA.
So, as I said before, anytime you hear the term gene expression, that means transcription, okay? So that means going from DNA to RNA. Basically, you're building an RNA sequence using a DNA code, okay? And in a minute, we're going to practice how to do that, making you in charge of being able to build RNA just by looking at a DNA code. Now, when we talk about gene expression, most genes in a cell are constantly varying expression. So sometimes certain genes will be turned on, whereas other times they'll be turned off. So we usually say activated or repressed, because think about it. If all of the genes in a cell were active at the same time, can you imagine how much RNA would be produced? That cell would basically explode, right? So you only have genes active when they're needed. Now, there are some genes that are always active. We call these constitutive or housekeeping genes. So circle those terms, constitutive and housekeeping genes. They mean the same thing. They simply mean that a gene is always active, okay? So it's always being expressed. These are basically genes and products that would need for processes that are always occurring in a cell. So think of life processes such as, genetic processes themselves, where you always need enzymes to be made for those processes. So for instance, polymerase genes, the genes that are responsible for ultimately making polymerases, those are always active because they're something that the cell will always need. Now, when we talk about DNA coding for RNA, there's actually all different types of RNA that's made. There's mRNA, rRNA, and tRNA. But the one that will ultimately encode for a protein is mRNA. So you're going to hear more about that in later slides. Now, keep in mind this process of gene expression. To express DNA, DNA is double-stranded, right? So those double strands are going to need to separate, just like we saw them do in DNA replication. Now, since DNA is double-stranded, but it's coding for RNA, which is single-stranded, you're going to end up with different types of strands. You have the antisense strand of DNA and the coding sense strand. Okay, now what you're going to notice in this process while RNA polymerase is building the RNA, these two strands of DNA have different roles, because remember, since DNA is double-stranded and it's producing a single-stranded product of RNA, only one of those strands is going to be the template, okay, the template or code used to build that RNA. Okay, so in the next slide, I'm going to demonstrate what this means, but for now, you can think of the coding strand or the sense strand as having the same sequence as RNA, except that, of course, DNA always has a T wherever RNA would have a U. So when you see sense strand, I want you to underline the S at the beginning of sense strand and write same. Okay, we're going to go over that again in the next slides, but I want you to start associating coding or sense with being the same code as RNA. Whereas the antisense strand of DNA is actually the strand that will be the template for building the RNA, which means that as the cell reads the antisense, it's going to pick the complements of that sequence to build the mRNA. So for antisense strand, you can make note that this will complement the RNA sequence. It will be anti it, so it won't be the same letters. It'll be the opposite, or the complement. Whereas the sense strand will be the same sequence. Okay, so we're going to visualize that now in the next slides, because you have to be very comfortable with making RNA simply by looking at a DNA code, the same way that a cell would. Okay, so to help you visualize the concept of the sense DNA versus the antisense DNA and how it relates to the RNA, let's think of it this way. Whenever you're given a DNA sequence, for instance, if you're doing any research and you look up DNA sequence online, you always find the sequence written out as a single strand in 5' to 3' direction. So when I say 5', that's 5' to 3' direction. And like I mentioned earlier, that's basically just a label of orientation, kind of like saying left to right, or top to bottom, or even north versus south. Okay, so 5' and 3' is just the orientation of DNA. Now, when you find this sequence, you notice this is a single strand, right? But what do you know about DNA? DNA is double-stranded. So even though you have this single strand, the DNA itself is actually two strands. And the second strand of DNA is always the complement of the first. Okay, and remember, for complements, we previously said you can use the memory trick at Georgian Court. The complement of A is T, the complement of G is C. Okay, now when we talk about 5', 3', those are the complements to each other. So the complement of 5' is 3', and the complement of 3' is 5'. Okay, so you can kind of think of them as opposites whenever I say complement. So if our DNA sequence is 5', C, A, C, G, G, C, 3', then the double strand of that DNA would be the complement. It would be the complement of 5', which is 3'. The complement of C is G, A is T, G, C, C, G, 5'. Because again, Each of these is the complement of the one we see above. So the complement of 5' is 3', the complement of the C is G, complement of A is T, complement of C is G, and so forth. Okay, right down to the very last number, the 3' complement is 5'. So now, if we're going to build an RNA strand from this, what did we say about any kind of polymerase? What direction do they always like to build in? 5' to 3', right? So our RNA, the RNA that's going to be made, you want it to be made in the 5', 3' direction. How would you get an RNA sequence that's 5' to 3'? Well, from this complement, right? So this strand here has to be the template so that our final result is 5' to 3'. Okay? So the way that you would do this is simply take the complement of each piece of this strand. So if we were to make RNA from this DNA, we would have to go step by step. Complement of 3' is 5'. The complement of G is C. The complement of T is A, G, C. C complement is G, G, C, and then again the final complement is 3' to this. Now when you look at this RNA strand that's been created in 5' to 3', what do you notice? What does this look like? This looks like our very first sequence, right? That's CAC, CAC. And you notice the 5' and 3' orientation is the same. So this strand of DNA would be called the sense, and this strand is the antisense. So you notice that the RNA has the same sequence as the sense strand of DNA, and it is anti or complementing our antisense. Okay, so now to kind of practice this a little bit more, okay, I'll have you do some practice problems. Okay, so to practice this concept a little bit more on sense versus antisense DNA and how to get the RNA that would be created during transcription, the way that you would encounter this as a question on an exam or whatnot is you'd first know whether you're given sense or antisense DNA. Okay, and it would always be given to you in the 5' to 3' direction. So in our first bullet point, we're looking at sense DNA. And like I said before, as soon as you see sense DNA, look at that S and remember same. So when you're given a sense DNA sequence, your RNA should look identical to that. Same number, same letters everywhere, except since it's RNA now, any T should become a U. Okay, so if we're given sense DNA 5', C, A, G, T, 3', then we have to make the 5' to 3' RNA. Since this is sense DNA that we are given, our RNA should look the same. So it would be 5', C, A, G, and then because it's RNA, U instead of T. And it continues to the 3'. Okay, so notice the DNA and the RNA look the same everywhere except for when you have a T in the DNA. Now it's a U. Other than that, it's the same. The other way you may encounter this is if you see antisense instead. And remember, the moment you see antisense, anti tells you that you should be complementing everything, including the numbers. So if you are given an antisense DNA sequence that is 5' C A G T 3', then you go one by one and complement each of these, okay? So it's not going to be the same number or letter anywhere. It's the complement. So when you ask yourself, what is the 5' to 3' RNA, right? Well, don't worry about orientation yet. First, simply complement everything, including the numbers. So the first thing you would do is, instead of a 5', the complement is now 3'. Then G instead of a C, the complement to A is U, C complements the G, A complements the T, and then 5' complements the 3'. Okay, so for each number or letter, you're doing the opposite, the complement. Now, keep in mind, the question will always ask for the final answer to be written in 5' to 3'. So if you give me this, then you are currently incorrect, because that is not the 5' to 3' RNA. So the last thing you always have to do is make sure to rewrite the final answer 5' to 3', simply reading this from right to left. So you'd write 5' A, C, U, G, 3', just like you see here at the bottom of the slide. So 5' A, C, U, G, 3'. Okay? So once again, anytime you're given sense DNA, you simply write the same numbers and letters exactly the way they are and replace any T's with U. If instead you're given antisense, then every single number or letter must be the complement, and your final answer must be flipped around to be 5' to 3'. Okay, so I always recommend doing the antisense in 2 steps. That way you don't accidentally miss a letter or write anything incorrectly if you try to do it in 1 step. Okay, so now I want you to practice this concept, because again, this is something that students usually get some points off during the semester for. So I'm going to give you some extra practice now. Okay, so just to make sure that you're grasping that concept and you're very comfortable with it and would be able to tackle any kind of problem that I could give you with it, I want you to stop everything right now and send me in the Remind app your answer for practice problems number 1 and number 2, which you will find on this slide and the very next slide. And please get me those answers by Thursday at midnight, because then the following day you have to start the next lecture or next lessons that we do. Okay? And here is the second practice problem that I want you to stop everything, pause the video, make sure you send me your answer as soon as you get to the slide. That way you do not forget to do this, okay? And then once you finish taking care of that, you can continue the video, and we're now going to head into some of the details of the transcription process and the various steps that are involved. So as you saw from the video, with transcription, just like you'll see with translation, you have the 3 terms: initiation, elongation, and termination, and you have to know what's going on in each of these steps. So initiation is how things get started. So with transcription, there's 2 key players in prokaryotic initiation. The first one is the promoter, and next to the word promoter, I want you to write DNA sequence. So the promoter is a DNA sequence that basically tells the RNA polymerase, hey, I'm a gene that needs to be expressed, come here and bind to me. So the promoter is what gets recognized by RNA polymerase in order to be expressed. Every single gene needs a promoter in order to be expressed. Otherwise, RNA polymerase, which builds the RNA, would never be able to arrive at that gene to transcribe it. Now, the RNA polymerase that's responsible for actually transcribing or making the RNA is made up of two subunits, and there's a trick to remember which subunit's doing what. For sigma, I always write sigma sees the promoter. So I underline that S and think sigma sees the promoter because that's the subunit that actually recognizes and binds to the promoter to start transcription. Whereas the core, that's more involved with elongation. The core constructs, okay, that's how I remember it, core constructs in order to build the rest of the RNA. Okay, so sigma sees the promoter and then the core constructs, and the core constructing is more the elongation step. So just to make sure that you have the memory trick written down in your notes, I've written it on this extra slide here. So sigma sees the promoter, and so it starts transcription. That's part of initiation. So whenever you're asked about initiation of transcription, remember sigma subunit of the RNA polymerase and the promoter. Okay, those are the two things involved in initiation of transcription. Whereas elongation, core constructs the new RNA strand, and that is elongation. Okay, so this is just another recap of initiation and elongation with a couple of extra details. So basically what happens is sigma sees the promoter of the DNA, it binds, and the core enzyme will then open up the DNA. 6 to 9 bases will be synthesized while at the promoter, and then at that point sigma is no longer needed, and so it pops off, whereas the core continues elongating the new RNA strand. Okay? And the reason why I write RNA pol is the rock star is to once again emphasize that RNA polymerase does not have the limitations that we saw with DNA polymerase. Now, with everything in life, everything that has a start needs to have an end. So for transcription, termination occurs when the RNA polymerase hits what's called a hairpin structure. And this figure right here shows what a hairpin loop looks like. And basically what happens is the RNA polymerase gets destabilized by that hairpin loop, and it causes it to pop off. Okay, so when you think about transcription, remember initiation is the sigma subunit and the promoter. Elongation is the core subunit of RNA polymerase. And then termination is the hairpin structure that destabilizes RNA polymerase. Now, when you go through all of this, students a lot of times just think of RNA as mRNA because that's what they know codes for the protein. But always remember that tRNA and rRNA are just as important. They all have a role in translation, which is where we're now going to make the protein. Now, to remind you with translation, again, you're going from nucleic acid to protein. And in this process, mRNA is the code that will basically tell the ribosome what amino acid needs to be where. rRNA is very important. That's actually part of the ribosome. And the fact that the ribosome has some rRNA, which is nice, clingy single strands of RNA, that allows the ribosome to grab a hold of the mRNA to then use it as the template. You then have tRNA, which is going to be what's actually holding the amino acids and brings them together so that the ribosome can build the protein. Now with tRNA, sometimes you'll hear it referred to as charged or uncharged. Charged simply means that the tRNA currently has an amino acid, whereas uncharged means that the tRNA gave away its amino acid, so it's no longer holding any amino acid. Now, one other thing I want to remind you of on this slide is the fact that this ribosome structure over here, which like I said, that has some rRNA on it. So this ribosome structure is what we call a nucleoprotein. Nucleoprotein is important because, again, the fact that the ribosome is protein because it's an enzyme, but also has some nucleic acid, the rRNA, allows it to grab to grab mRNA, which is important. Now, the ribosome has 3 sites on it. It has A, P, and E site, so you could remember it as APE or EPA. The A site is the amino acid acceptor site, so the tRNA that's in an A site will have just a single amino acid. It'll be charged. The P site is the peptidyl or peptide site, so the tRNA that's sitting in the P site will have the growing polypeptide string, the growing protein, and the E site is the exit site. So that's where the uncharged tRNA will then leave the ribosome. This will become much clearer in a minute when you review the video. Okay, this slide is just so that you can pause the video and write down the notes in case you didn't catch what each of the sites were. So now just to go over the 3 main steps of translation before you visualize it in the video. Just like transcription, there has to be an initiation, an elongation step, and a termination step. So with initiation, what we have is the ribosome being in charge of the grabbing hold of the mRNA. And the way it does that is mRNA will have what we call a Schindel-Garno sequence on it. And that complements the piece of rRNA that's part of the ribosome. So like I said, complements, single strands, they're very sticky and want to match with each other. So this part here, this is the ribosome, and it grabs a hold of the Schindel-Garno of mRNA. That's part of initiation. The other part of initiation is the fact that the mRNA has a start site that matches the tRNA that will bring over what we call the initiator tRNA, which is FMET. Okay, FMET will always be the first amino acid in any protein built. Now, what happens is, before I put the bullet points out for you to see, I kind of want to help you visualize what's happening in the process, and then you'll get to see it again in the video as well. So when you have translation occurring, first of all, the ribosome grabs a hold of the mRNA at the Schindel-Garno. Then picture a tRNA holding the first amino acid, FMET. It's going to come into the ribosome. This will be the only amino acid, the only tRNA that enters the ribosome at the P site instead of the acceptor A site. So the tRNA holding FMET comes in at the P site. Now the second charged tRNA holding a second amino acid will come into the acceptor site. And then I want you to always make the sound pop, kind of like a popping sound, pop, to remind you that the transfer will be the amino acids being held at the P site will transfer to the tRNA in the A site. So that's why I say pa, because the transfer is from the P site to the A site, which you'll see in the video in a minute. Once that occurs, the ribosome then shifts, and all of the tRNA ends up in exactly the right position for what we explained the ribosome sites earlier. So once the ribosome shifts, the tRNA that gave away its amino acids and is now empty and uncharged, that will be at the E site to exit the ribosome. The tRNA that just took the amino acids and is now a growing protein chain, where will that be? That will be in the P site, the protein site, the polypeptide site. And now the A site is open so that the next tRNA can come in, sit at the A site with its single amino acid, and then transfer can once again occur, pop, from the P to the A site. And that will keep happening over and over, which is called elongation. Okay, so these bullet points here show what I was just saying, the P to the A site transfer, and the shift in the ribosome. Then you can't have this go on forever, so termination has to occur. And what termination is when the ribosome reaches a stop codon. And I want you to circle stop codon and remind yourself that there is no tRNA that can read this, which means no amino acid can be brought in to the ribosome once you have a stop codon. Instead, release factor protein, sometimes you'll see it written as RF protein, that binds to the stop codon and allows for the protein chain to be released and the ribosome to be disassembled. Okay, so make sure you repeat this slide once or twice to kind of help you visualize the process of translation. To further help you visualize it, here we have on the next slide a nice little video.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. An aminoacyl tRNA bound to elongation factor TU, EFTU for short, enters the free A site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. Elongation factor TU hydrolyzes its bound GTP and dissociates. If the tRNA is correctly matched and remains bound for a long enough time, it is committed to be used in protein synthesis. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformational change. Elongation factor G binds to the ribosome. Hydrolysis of GTP by elongation factor G switches the ribosome back to the state in which it can accept the next incoming tRNA.
Okay, so that was quite a bit of information. So now you can either take a break, have a little snack, or you could head on over directly to the second half of the lesson. Okay?